Acute inflammation is caused by tissue injury, which can arise from physical damage, pathogenic infection, and lifestyle factors. Depending on how the injury occurred, a corresponding danger signal, DAMPs, PAMPs, or LAMPs, are released, which bind to toll-like receptors. One of the types of cells that toll-like receptors are expressed on are macrophages. Macrophages are a type of white blood cell that are part of the immune system, present in virtually all tissues in the body. Their roles include removing dead cells and debris, as well as releasing cytokines when needed. One of these cytokines that macrophages release is tumor necrosis factor alpha, or simply TNF-alpha. TNF-alpha is a pro-inflammatory signaling molecule that, depending on what conditions it is released in, can have a variety of different effects. Let's take a look. When the body sustains some kind of tissue injury, acute inflammation immediately occurs. As mentioned earlier, DAMPs, PAMPs, and LAMPs bind to toll-like receptors on a number of cells, including macrophages, which subsequently begin releasing cytokines, including TNF-alpha. TNF-alpha, being a pro-inflammatory signaling molecule, upregulates adhesion molecules so that circulating leukocytes and immune cells slow down and adhere to vessel walls, contributing to chemotaxis. TNF-alpha also increases the permeability of blood vessels in order to allow for greater migration of other immune cells, including more macrophages. Macrophages phagocytose foreign particles after opsonization and chemotaxis, helping to eliminate the irritant and eventually restore the body back to normal. This inflammatory process is what is responsible for the 5 classic signs of inflammation: rubor, or redness, caused by increased blood flow to the affected area; calor, or heat, the affected part is warmer due to the influx of blood; tumor, or swelling, from the accumulation of fluid in cells; dolor, or pain, caused by the release of chemicals that stimulate nerve endings; and function lesa, or loss of function, which results from the last 4 signs. This is all great in acute inflammation, but the homeostatic functions of macrophages and TNF-alpha can quickly become detrimental. If the irritant or trigger cannot be eliminated, or otherwise has a prolonged duration despite not being very intense, or the initial cause of inflammatory response was an autoimmune reaction, chronic inflammation occurs. Macrophages remain persistently activated as a result of continuous exposure to DAMPs, PAMPs, or self-antigens. This means that they continue to secrete pro-inflammatory cytokines, which perpetuate the immune response, continue to release enzymes like matrix metalloproteinases that break down the extracellular matrix, which contributes to tissue damage and scarring, or fibrosis, generate reactive oxygen and nitrogen species that cause oxidative tissue damage, and continue releasing more chemokines which recruit even more immune cells, including macrophages, to the site of inflammation, making it worse and worse. TNF-alpha, which continues to be secreted by macrophages, continues activating endothelial cells to increase permeability, thereby allowing more immune cells into the site of inflammation and really just perpetuating this cycle. However, despite their ostensibly destructive natures, macrophages and TNF-alpha also play significant roles in wound healing. After the inflammatory phase, macrophages transition from a pro-inflammatory phenotype to a tissue-repairing phenotype, now secreting anti-inflammatory cytokines such as interleukin-10 and TGF-beta, various growth factors, and ECM components to begin rebuilding new tissue. While TNF-alpha is predominantly pro-inflammatory, it does also contribute to the proliferative phase by promoting angiogenesis, which is needed to deliver oxygen and nutrients to the newly made tissue, as well as activating fibroblasts to produce collagen and other matrix proteins to help the structural integrity of the new tissue.